Hello, everyone, and welcome to today's SSP webinar, Linked Databases, Publishing Research Data. I'm Jeff Lang with the American Chemical Society and the chair of the SSP Webinars Working Group. We're pleased that you could join us today. In a moment, we'll be getting started and hearing from our moderator and panelists. You'll be seeing the SSP splash screen during this webinar since this is a discussion and there will be no slides. Your phone will be muted automatically in consideration of our presentation, or our presenters and your fellow webinar participants. Please use the questions feature to send in any of your questions. Or if you need to let us know that you're having any technical difficulties, you can use that same section. The moderator will review these questions and present them to the panelists. To help her, please specify to which presenter you'd like your question directed, and also please send in your questions as we go instead of waiting until the end. At the conclusion of today's presentation, you will receive a webinar evaluation via email. We encourage you to provide feedback so we can continuously improve our webinar program, and you will also receive a link by email to the recorded broadcast of this webinar. Our moderator today is Jennifer Landsberg. Jennifer is the product manager of the Cell.com publishing platform at Cell Press and brings a customer-focused, data-driven approach to her work. Prior to this, she was managing editor of the journal Neuro, where she oversaw production and operations for the journal. In this role, she led an initiative to encourage authors to identify the key resources used in the course of scientific research, ensuring reproducible methods and providing critical data to help researchers identify suitable reagents and tools. Here is Jennifer, who will introduce the panel and get us started.
Thanks, Jeff. Our speakers today, first is Walter Hawk, the VP of Research Data Management at Elsevier. Walter is responsible for research data management at Elsevier, specifically the Mendeley platform. This is an open ecosystem of researcher data tools, a data repository, an electronic lab notebook, a data search tool, and a data project management tool. Aside from his work for Elsevier, Walter is part of several open data community initiatives. For example, he co-chairs the RDA WSX Scolex Working Group on data article linking. He is part of the JISC Data to Paper Advisory Board, and his group participates in the NIH Data Commons Pilot Project. It is all about the RR FAIR data, focusing on data reuse. Prior to Elsevier, Walter has a background in online product and strategy roles. He has worked at eBay Classifieds in roles varying from business development to overall responsibility for the classified businesses in Italy, France, Belgium, and Turkey. Furthermore, he has worked for the Boston Consulting Group. Our next presenter is Anita Vandross, Principal Investigator, Ucsd, and founder and CEO of SciCrunch, Inc. Dr. Vandross started her career at the bench studying auditory neocortical neurons. However, during a postdoctoral fellowship at Stanford, she discovered high throughput biology in databases and worked on the Panther database for several years, contributing to the emerging field of genomics. Dr. Vandross brought her passion for neuroscience and the new data-intensive tools to bear on neuroscience data, while contributing to and later leading the Neuroscience Information Framework Project, a blueprint for neuroscience NIH initiative. While bench science is more like a cottage industry, the omics era requires rigorous standards to combine and make sense of data being aggregated, leading Dr. Vandross and her colleagues to lead efforts in the development of ontologies and data standards. Currently, Dr. Vandross leads the RRID initiative, a standard for publishing reagents, and she has become a founder and CEO of SciCrunch, the technology developed under the Neuroscience Information Framework in order to serve the scientific community and improve how we communicate about reagents. Finally, we have Justin Cherney, the VP of Operations at Jove. Dr. Cherney's path to passionately working on reproducibility started with an interdisciplinary PhD in biology and computer science, focusing on proteomics and quality assessment of peptide searching algorithms. From there, he joined Jove, the first and only peer-reviewed scientific video journal, whose focus is on increasing reproducibility through video publication and methods. As the VP of Operations, he's responsible for Jove's editorial, video, and IT teams, and is continually motivated by Jove's mission statement, which coincides perfectly with his passion for interdisciplinary work, innovation, effective integration of new technologies, and the reproducibility of science. So to get us started, I would ask our panelists, can you talk broadly about what the mechanisms your groups use to provide links to data in your articles?
And who would you like to start?
Let's start with you, Walter.
Okay, well, I can start. So I think your question is about how do we provide links to data in general, and also how do we provide links to data in articles. And the way we do that is actually changed dramatically in the past 5 years. In the past, as Elsevier, you know, we run like 2,500 journals, and we would have individual kind of setups between ourselves and the individual journals and individual repositories. So you can imagine what a nightmare it was to maintain, let's say, 1,000 times, let's say, a couple of hundred relationships, and each relationship was different, in order to link data to articles. So every repository would have their own standard, every journal would have their own standard. So it was actually very hard to technically link articles to data sets. And what happened is, of course, we encourage authors to cite data sets, we encouraged authors to talk about data sets, but then in the background, technically, it was a complete nightmare. In the past couple of years, we have shifted from that many-to-many situation to a very much, I would say, easier framework that is called Scolex.org. And the Scolex Initiative, which is spelled SCHOLIX, the Scolex.org initiative is very much trying to streamline the data to article relationships. And it has worked. So instead of now having this 1,000 to hundreds relationships, we actually have two big hubs, namely Crossref and DataCite, that already communicate on a daily basis through APIs with the repositories and the publishers. And the only thing we need to ask to repositories is please add any information you have about datasets to articles to that framework. The same goes for ourselves. All our journals add that information to Crossref, and then it gets automatically picked up by the platforms that consume that. So for example, you now find all these links on ScienceDirect, but you also find them on Scopus and you find them on Mendeley Data. So for us, actually, we've gone from a pretty complex many-to-many environment to a really straightforward kind of hub environment, where we agreed on how to share this information. And then it's really helpful to have then parties like DataCite and Crossref and also OpenAir and ANDS in Australia also played a big part supporting that to get big adoption. At the moment, it's big contributors like DataCite and Crossref who are contributing links, but also individual publishers like Springer Nature, Elsevier, and IEEE. So the first thing I think is it has helped to standardize this process. Thank you.
Thanks. Anita, what are the difficulties in getting authors to publish data, and what incentives do you think we can provide to make it easier?
So I think one of the really important things that's often missed between a lot of these initiatives of how to standardize data, and all sounds very good, right? But one of the things that we found is that you really need a driving use case that resonates well with authors. So we have about 9,000 articles currently, and many of them are in the Cell Press journals. Thank you very much, Jennifer. That's it's certainly not all of them, but it is, there's quite a bit of this adoption now. And we found initially that there was a kind of a driving use case for authors. Everybody was very frustrated trying to find antibodies because of the way that they were cited. And it was one of our kind of best drivers in terms of, why would I do this? Why would I take extra time to actually look this thing up? Because that was really the question that we needed to solve. Why would any one individual author do this out of the goodness of their heart? Well, two things. One, they had to have been frustrated by this. And two, there had to be a relatively easy way for them to actually find these identifiers from the database. And then there had to be a very unambiguous way to kind of get them in. And, we find actually that the identifiers put in by authors have an accuracy rate of around 97 to 98% if you just don't worry about the syntax errors, which are going away as journals link those objects, those identifiers, into actual linked data objects, which are wonderful, right? That's a really good rate. I mean, we'd like to go higher than that, but coming straight out of an author who's doing this without any help, without any linking, they're already at 97% accuracy, which if you compare that to, basically what you can get out of the author without this initiative, which is about, at best 50% accuracy, that's kind of the best case scenario. We've gone a huge way towards findable reagents. And so now the question is, how can we, take that data, how can we make that data more available to people so that they start to actually use it? And for the publishers, the use of that data is really important. For some of them, we used to be one of those, hundreds of data providers with all of our own stuff. And we're very excited about the Scolex standard, especially if we don't have to change too much. But essentially, we have this idea that you can actually take users from some content to some published content, some articles to other articles, back to other articles, and using the reagent as a link between those articles. Because a lot of times we find that authors are actually trying to nail down their protocols, and they're trying to figure out why something's not working. So they'll go to papers, and if there are easy links to other papers that also use that exact same reagent, then they can actually, do that work, and they do that within the publisher's website. So long story short, I think that you need a really good driving use case, and then we're actually trying to add more things to be more relevant to the authors.
If I may chip in for a second to what Anita just said, I think you're spot on, right? It is really about finding a use case, and I really love the way you express that through, for example, reagents, and finding the same reagent in different papers or different protocols, and how is that then applied in different experiments. And then as a researcher, to get that at the tip of your fingers and then immediately see the different applications actually really helps you set up your own experiment. We actually see the same for actually many use cases. So we see the same, for example, in software, where there are certain algorithms used for different applications. That can, and this is software in the broad term. So this can be algorithms used, for example, in health science, where it applies to certain workflows to analyze certain genes or sequences. Like, if you can find those specific algorithms linked to different papers or different data sets, that is actually really easy. So what we're doing is actually, yeah, exactly. So I think
Jennifer actually has that in her star table, and a lot of those are starting to get RIDs. So that, again, we can provide that data, and we try to all of the users through what we were talking about offline, the Crossref event database.
Exactly.
But I'm sorry for interrupting.
No, spot on. I think interesting bit is, like, we need what you're doing with, like, making sure that RIDs or persistent identifiers get added to these relations is really helpful. I think secondly, what is also helpful is, I think you touched upon the frustration of researchers when they've tried to do it themselves. They may be then subsequently a good citizen and proactively share that in their next article. I think also what helps is perhaps to educate authors a little bit about the fact that this actually helps in terms of their impact or their success. I mean, I'm sure actually, Jennifer, you can tell of your own experience how the star tables have helped the cell journals, not just in terms of reproducibility, but I think also in terms of people then trying to seek it out and using it because it's really easy. Once you have a standard and people can recognize it, it actually becomes more easy to replicate stuff. And the second thing we're doing is we've now analyzed thousands of papers, actually millions of papers in different countries to see if it helps to, for example, add data to that paper, and we then compare that to papers where there was no data added. And actually, no matter how we cross-cut the analysis, it always turned out that by country or by domain or by institution, it always turns out that papers that have data sets attached get way more citations than the papers that do not. Of course, there's a question about causality, right? It could be that papers that are generally good generally also have more data attached, but it is kind of like what is interesting. Even if you say, well, the causality might be something different than correlation, even if you show that picture to researchers, they start to scratch behind their ear and they're like, maybe it actually really does help to add this information to my paper, because it could be that the causality is that only good papers have data, but if I start adding my data, actually it may help as well.
Yeah, exactly. And so I think that first of all, that's fantastic work, and I'm very glad to understand that, any way you sort of cross-cut that data, you still get the positive effect, which is something that would be absolutely amazing to try and show with, with our IDs as well. But you always run into this, correlation versus causation bit. But on the one hand, I actually don't care because if it drives, if this is something that isn't too hard to do, but it drives good behavior, right? Then you want the literature to include data. You want the literature to be, less ambiguous. You want to drive that good behavior any way that you can get that good behavior. And if there is a good use case for that, that good behavior kind of driver, I think it's really, really excellent.
So we have a couple of questions from our listeners. One was briefly, can you spell the name of the data sharing initiative again? I believe they were referring to Scolex. So that is SCHOLIX.org. Perhaps we could put this in the chat. And then there was another question on where do authors find the links that they need?
So one of the things I can try to address that from our side. So when we work with a particular journal, including Cell Press, there is a set of instructions to authors that link out to one website, and this was really important to us to aggregate, the 20 or 30 databases, all under one kind of look and feel, all under the same functionality. Into one place, so that it makes it as easy for authors to look up these identifiers as it possibly can be. Now, if you're searching with an ambiguous term like GFAP antibody, you're still going to get hundreds or thousands of different reagents. But the point is that at that point, before you publish the paper, you want to, hit the author with, hey, this is ambiguous. And we've had, we have a help desk that really helps authors try and figure these identifiers out, and we're on there every day. There are now about, I would say about 12 help requests a day, various ad requests or excuse me, just help requests. And so if we make it as easy as possible, but it can't be easier than it has to be. So it has to be a little bit hard, especially for labs that don't keep really good notes. If they don't keep great notes, then they're just going to face that ambiguity that's going to need to be addressed. And so they're going to need to go out, they're going to need to find the reagent bottle. They're going to need to go out and find these things. But once they do that, you want to make it just as easy as possible for them to be able to comply with the standards. So our answer to this has been, you run one website, You ask these authors to come to exactly one place, and then you have a help desk that helps them actually complete that task if they are confused in some way.
So it's interesting. Oh,
sorry. We also try to keep the journal editors out of this whole process if we possibly can. I mean, sometimes editors will get into some of these interactions with, like, for example, you know, a stock center or one of these, but you know, mostly we try to keep that away from the workflow of the journal editors.
What about on the review side? So from at Jove, we actually make it's part of the reviewer's tasks to ensure that any resources that we have are appropriately linked or appropriately identified, or there's information surrounding the link that allows that to be discoverable. So I'm curious from your side if that's something that you guys can kind of do. Is that something that's an option?
You mean like, so we work with authors before they publish, right? So we're dealing with, can I please have an identifier for an antibody? Can I have an identifier for an animal or a cell line? And then that's when they come to us. The URLs to reagents are incredibly unstable. Because of search engine optimization. Sorry, Google, but you have totally royally screwed us up in terms of the academic enterprise, because you know, the Google algorithm will change, and so antibody companies will change how their links look so that they can get higher ranking. And that has just destroyed our ability to have journal articles link over the long term to the same reagent. Now, sometimes the antibody companies will actually leave nice forwarding addresses, and many times they won't. Does that make sense, Justin? So we like persistent identifiers because links break. That's that's one of the kind of driving use cases. Now, I know that your data is incredibly good and can actually, you know, people can go back to that within a certain period of time and absolutely find the right identifiers. But without the identifiers there, in 2 to 3 years, you will have, you know, a quarter of your links will break. So we've actually tested that across the entirety of the open access literature, the accessible literature to text mining. We looked at all of the links, which was, you know, many hundreds of thousands. And within, you know, roughly 2 years, a quarter of them were broken. So that's why we like to not have links per se. We like to have the identifiers because the identifier can stay stable. The links can be left as a set of, you know, alternate URLs or old URLs. So example, like Walter mentioned, the idea is that you would have, you know, some of these scripts and these tools. Well, you know, a lot of papers that I've actually reviewed, you know, as with my reviewer hat, have things to have links to various code repositories that basically no longer exist or are being decommissioned. Like, everybody thinks that, you know, GitHub is the way to go right now, but, you know, Microsoft took it over, and right. At some point it's not going to be cool anymore. So there will be the next repository of code, and then everybody will move their tools there, and the link structure is going to change. So I think that there is definitely a reason to have a repository of these things, which provides identifiers, which stays over the long term, and it allows all of those URLs to be aggregated to the same project, as long as the code, you know, we can agree that the code is reasonably the same. As long as the antibody is reasonably the same antibody, then we say, okay, well, that's great. This is an antibody, this is the same thing. But it's now being offered by, you know, Millipore, because Chemicon was just bought by Millipore, or rather it went to EMB and Sigma and whatever else it's going to be tomorrow. We don't know, but that whole catalog of reagents went, you know, through all of these various companies to now end up wherever it is. So that provenance is just, it's really hard to do when you're publishing a stable object that is immutable. All of these web-like things will just flow around that object, and eventually the links will unfortunately break.
Yeah, it is a problem that we do try to entertain, and we do try and solve. In fact, we have 404 checkers that we run on I think we run it every 3 days to check to determine if any of our links are in fact not working anymore. But I recognize how that is not a stable solution to that problem. And I think that that's something that we're in the process of trying to address as well. The permanent linking ID, though, that's a great solution to that problem.
Well, you're welcome to use our IDs whenever you like. It's free and open, and you can just send people our way. We will try to do our absolute best to provide them the identifiers that they need.
Yeah, I mean, if I may jump in, I mean, I really agree that using persistent identifiers is the right way to go. The way I kind of look at that is from a both a publishing or a data perspective. I don't think it's only RRIDs, right? There's actually many persistent identifiers out there that are good for different purposes. So there's data center IDs, which are usually run by DataSite, and then there's even GenBank, although it's not perfect. You can actually resolve them quite well. And each domain has their own standards to kind of run this stuff. What we do is we actually look a lot at identifiers.org, which is a site that kind of looks at what are the persistent identifiers out there, and we try to see if we can use that site to resolve a persistent identifier, which is for us a criteria to use the link or not. If we can then not resolve it, so if it looks like just a web link, we generally do not accept it. Mm-hmm. So we actually coach authors to use some form of persistent identifier, but it doesn't really matter which one they use. They probably know best which one to use. And if they really don't know, we then help them. We tell them, well, why don't you use this one or that one? But it's really, but I see your point, because not all suppliers of chemical reagents will obviously, or bioreagents will have a use of a persistent identifier. Mm-hmm. So that's an interesting challenge. I mean, we should maybe even work with them to see if we can develop a schema that works for them, or be adopted by them.
Yeah, and actually, this is one of our big outstanding issues. So we've got this quote unquote mandate from the National Institutes of Health, right? They're really working on, well, here are the reagents that are most variable in most experiments, which means that these are the things that fail most of the time. And there are some in the biological and there's some in the chemical realm. And with chemicals, it's actually relatively difficult to understand, what is the best way forward. So for biological entities, we know they're antibodies, they're organisms, they're cell lines. These are really the key players in this portion of the mandate for increased rigor and reproducibility. But once we have, once we go into the chemical realm, I still haven't done a full analysis, and maybe you have, Walter or Justin. What is in PubChem and how far we are from that, with that database compared to where we need to be to really address every single chemical that would be needed. Because one of the things about persistent identifiers, it's great when one author does just a fantastic job and identifies everything in their paper. But what you really need is you need every author to take at least one small step together. And I think at that point, you really get a much broader coverage, you get much more adoption, and you can really ask, all the authors then to take a second step once they're sort of comfortable with the first one. So, for us, we've been struggling with with adding chemicals because, again, we don't really know yet what the coverage is for some of these databases. And if every, chemical that some author has to add to their paper is basically not available, or any type of object, right, is not available, if there's not enough coverage in the database, then the problem becomes, well, I'm not going to do this, right? So then you get the authors pulling back, they bulk a little bit. And if you have enough coverage, it's a lookup. If you don't have enough coverage, it's a real problem where the author can't find what they need to register. Well, registration is a longer process. They may not do it. So I think, again, it's great that you're going with identifiers.org, and I wish one of the things that was listed in identifiers.org are things like, is there a help desk? What is the percentage coverage of a particular field or type? And it's great when the database is actually already the standard for a particular subfield. That's fantastic. Everybody already knows it. Then your job is done. But how do you get to that place where you can tell, and from a journal perspective, there are thousands of databases, right? So which one do you go with? How do you know that's the right one? Presence in identifiers.org certainly helps.
No, but I take it the other way around, because I think you're spot on, right? Like, not all databases have full coverage, and then there's also the question which databases to promote. But I mean, I think actually there we usually ask the editors and the publishers and the community to come up with what they prefer, what do they think is right, and they come up with a proposal, and we then actually suggest in our submission flow to use those repositories as a kind of go-to place. Now, obviously that's not perfect, but I think the biggest challenge we have is author uptake. So it's not so much the question like, do we pick the right repository? Because I think that's actually in most domains pretty straightforward. There's like a couple of them, and sometimes there's none, and then you may recommend a general repository. You can always recommend one or two, and the key challenge for us has been author uptake, which was, let's say, less than 10% in many journals, and we've been able to increase that a lot by just proactively reaching out, explaining, educating to way higher percentages, and that's the key point. I think you made that at the beginning as well, which is if only the sporadic author goes through the effort, it doesn't help the domain, it doesn't help the field. But if you persistently, in the same way, very simply, with maybe also the same data sharing policies, expose this to authors, the uptake is actually a lot higher, and we see it now increasing a lot. So I think you're right that the standards and the coverage, and that's all a problem. But I think the biggest challenge we all collectively have is author uptake and journal uptake, and that's where we can actually make a big difference.
Yeah, I agree, actually. We find that through our own publication process, which includes the production of a video, which is outside of the normal bounds, right, is that author involvement is one of the things that's really hard for us to kind of work with. I mean, with enough education and careful conversations and well-structured email and things like that, we tend to see some uptake. But one of the issues we have too is even in reproducibility as a subject matter and working with authors, they seem to be very initially excited, but then when you start asking them to actually do things, you tend to get a little bit different of a response. So we have found that through very careful education and saying like, this will make your article better. That it seems to be a strategy that's worked. But I completely agree with you. I think that is the biggest challenge that we face with this, is that we have good intentions. We want this to be reproducible. We want to do things, but sometimes getting author involvement. I agree. I think that's a very big challenge.
Yeah, and I think the solutions that are currently appearing, which is more along the lines that, for example, the Transparency and Openness Initiative from the Center of Open Science, which I really think is great. They've come out with 5 simple standards to say, okay, this is how you can share your data. And well, I mean, actually, as Elsevier, we've taken the step to basically follow those 5 standards and say, look, I mean, instead of having many different data sharing policies and how you talk about your data. We just do it in 5 simple ways. I know Springer Nature has done something similar. It's 4 simple ways, and actually our 5 match very closely with Springer Nature's 4. That standardization, I think, is actually very, very helpful. And we're now working together through the Research Data Alliance initiative to kind of see if we can harmonize them across all publishers. Well, that of course is a long process, which I'm sure will hopefully end in one standard. But actually, sorry, not one standard, but maybe 5 or 6 standards. Bring that clarity to authors, and then also indeed explaining to them that this is not about just making your paper more reproducible, but also about making sure that your paper has more impact. I think that are the key bits. And then the third bit is you need a very simple submission flow, a very clear submission flow where it says, in this part of the submission flow, you need to think about submitting your data and your identifiers and your related research. And this is why you need to do it. And I mean, I guess because Jennifer's online as well, I mean, the STAR method is one way, and then another way is just simply the data availability statement. Yep. And these efforts that I think are now happening more and more across publishers are simple ways to make sure that authors actually understand it. And I think it also helps that different publishers are now starting to do this in the same way. So the data availability statement is something that is not just a single publisher, but it's actually spanning across many publishers. And I think publishers are starting to recognize that this is actually in their benefit to talk to authors in the same way, to explain to them that this helps in the same way, because we're all striving for author uptake, and we don't want to educate each author in a different way.
Yep, exactly. And it makes, not that anybody wants to hear this, but it does make papers more portable, which is a real benefit to authors. I mean, as an author, one of the things I really, really hated is making sure that my, my references had the appropriate points and, or periods and commas and whatever the heck else. And, eventually you move out of, you move into a document format that, your linking into, your references doesn't work anymore. So half the stuff doesn't work, and I'm just having to, do this, reformatting essentially as a complete waste of time. And it's terrible if we kind of take that cottage industry of making sure that our references are in one journal format versus another to data, because that's kind of the kiss of death for data, and it's the kiss of death of identifiers. So the best thing to do would be to have one standard across all of these different publishers with regard to what we do with data, because data by itself, it's that one article, one author, it doesn't actually solve anybody's problem. Data and these reagent identifiers and these things really only work when there's a lot of people involved, and if they do it exactly the same way. So that's one of the things that we really again fought for in the RID initiative, because we really, really wanted to make sure that if somebody is going to one journal, they have exactly the same format as with another journal. Now they can put those identifiers anywhere they like. The STAR methods tables are fantastic, and they're actually being adopted by many other publishers, or at least they can go in if they, it might not be called STAR methods, but the tables are there and they're becoming more prevalent, which is fantastic. It doesn't really matter if the one column is here, another column is there. It's better if they're all the same, but at least the relevant pieces of information should be available in the same format. That way it's really, really easy. You don't need text mining to pull these things out. You can use Google, right? Every author, every reader can use Google Scholar to actually find these things. And when they're searching for these identifiers, and they can actually get back every paper that mentions that identifier, I think it becomes powerful. It becomes tremendously powerful. And if you have, for example, if you have catalog numbers and URLs, because the URLs changed, they may need to search for things that no longer exist just to pull them, all of the references out of papers. Whereas if we have an identifier, we have that standard way of either addressing data or a way of addressing these reagents. This is the way that we can now aggregate all of the references to that thing. And I'm not sure if anyone's aware of this tipping point notion, but if there is a tipping point, it's usually around 10% of the total papers. So we'd love to see this in 10% or more of the data, and then things will go faster, basically. And we'd love to see this in 10% of the articles in terms of the RIDs. If we'd love to get it into 10% of the articles, because then again, we're at a point where everyone has seen this, and everyone can see, there's some tangible benefit to actually doing this to them as they're trying to track down their reagents. And
what do you guess? Sorry to interrupt, but what do you guess the current uptake is? What's the current percentage?
So I've actually done a lot of work on this recently. None of it is published, but I was trying to really address with some of the stock centers. So for example, if you look at Jackson Laboratory, we're in the 5% roughly over the last year. If you look at the MMRC repository, this is another mouse repository, we're actually much higher. We're in the 20 to 30% range. So at least for some of our data sets, we seem to have, passed the tipping point. For Synaptic Systems, it's an antibody manufacturer in Germany. They're fantastic, actually. We are definitely beyond the tipping point. For other great manufacturers like, say, Thermo or BioLegend, we're not quite there yet, but I'm not sure if, if we can truly assess, because they have a much broader catalog, so which, not all of it is antibodies, so then it's very difficult to actually find out what the percentage of antibody uses versus, something else. Pipette tips, we have that problem with the bigger manufacturers, but certainly where we've looked and the antibody vendors are small, and the catalogs are very much just antibodies, or for the stock centers where the catalogs are just animals of a particular type, we're somewhere near the 10%, sometimes quite a bit higher, but we're kind of getting to that point with at least.
Okay, very good.
But certainly not all, and I would love to continue to move in the direction and kind of push everybody towards actually using more of these identifiers. That way, again, it becomes more relevant. It becomes part of everyone's workflow.
So what would you say, how important is linking data to reproducibility in general, and what more can publishers be doing to promote reproducibility? This is for anyone to jump in.
Oh, I would love to take that one. So again, I think we've taken a long, hard look at the reproducibility at the NIH reproducibility guidelines. We were in the room when they were decided. For some of our staff were in the room when these things were decided. Some of them weren't. And there are, in terms of reagents, there are kind of 4 major categories. We handle 3 of them. And we handle 3 of them very specifically because these are, again, driving use cases for things that people are very frustrated about. And this is one of the reasons why I think we have had not as much, pushback from authors, because authors have been frustrated by these things. These standards from NIH didn't come from nowhere. They came out of people's frustration in being able to reproduce work. So we're really meeting a lot of those. We're not yet meeting the chemicals, and that's one of the things that we are taking a long, hard look at and trying to figure out how to expand the initiative to do this well. And I would love to say I will have an answer for that. I think that this linking is the first step in reproducibility, because if you look at the whole reproducibility guidelines, there are things like randomization. How does is there a power analysis done for the number of subjects in the study? How many are the statistics appropriate that are being used? And there's a whole slew of other things. But I will say one thing: if you cannot find the reagents, you will not reproduce the study. You don't have a snowball's chance to actually reproduce that work. So while getting the reagents right is not sufficient for reproducibility, it's certainly necessary. And so I think that we can, again, linking these things to the databases which take care of these items is one, I think, very important, but certainly not a sufficient step towards reproducibility. It is, however, something that, again, authors tend not to balk at. We've had that once now. We've had exactly one author tell us to go away. And again, it was because he had a lifetime of work, apparently, that he would have to register. And he just said, no, that's just too much work. I'm about to retire. This is my last paper or some such. And we're like, okay, fine. But generally, especially younger authors are really, really excited about being able to link these things because they see that as an improving reproducibility. There's a tangible benefit for them. And they're able to really, showcase that also in their progress reports. There's been a lot of changes at NIH to show, here's my authentication of key biological and chemical resources document. How have I done this? How have I met this in the past? And one of the things that they can, they can really do is they can say, hey, look, here's my STAR methods paper, and it's all linked. And look at that lovely, set of identifiers. These things are all linked to the, to the authority that takes care of these kinds of, these kinds of objects. And there are things like, warnings from the ICLAC and other groups that can then be attached to those unique identifiers. Now, when I say ICLAC, what I'm talking about is the International Cell Line Authentication Committee. So I'm sure that you guys have heard about these problems with cell lines. And the problems with cell lines is that sometimes, people share them and then they end up being either contaminated, they might be misidentified, there might be other problems with those cell lines. And again, driving people to a place where they find their identifiers, that place contains all those warnings. So they can then decide, is this an important warning for my particular paper, or is this not an important warning for my particular paper? If they're using a hepatoblastoma cell, and they think they're using a hepatocarcinoma cell, maybe it matters to their paper. Maybe it doesn't matter to their paper, right? They just have to kind of understand that. But generally, one of the things that we've actually spent a lot of time on very recently as well, which should be published, I hope, at some point, is the, just really looking at across the literature, if you use a cell line RRID, where the person has to look up the identifier, generally in one website, and that website contains these warnings. We've looked at, what is the percentage of those papers that actually use cell lines with those warnings. And I will say that, as a correlation, correlations are never perfect, but certainly the use of the the problematic or contaminated cell lines, or at least cell lines on that list, is much lower. So, we've cut that in half, And then when you look at the reality, it's a much even smaller percentage of cell lines that actually need to be examined again, because a lot of those warnings, again, are not relevant to a particular paper. So whereas in the generic literature in using cell lines, it's about 10 to 15% of papers that have at least one cell line on this list. That percent is about 5-ish in the RID literature, which it's not gone, but again, the use of these things really improperly is almost gone. It's almost eliminated. So it increases the ability for us to say this is more reproducible, because at least the author is seeing these warnings and they see it at the time that they can do something about it. It's not later, after they've already staked their reputation on this. It's before they've published.
Justin, do you think you could also comment about what, especially coming from the perspective of Jove, like what more can publishers be doing to promote reproducibility?
Yeah, so one of the things from our side is that we really focus on methods. And I think that to all of this conversation has been really good about talking about reagents and talking about having the right reagents and doing that. And that's very true, and having access to all of the data that's available. I think that's also something that's really important, because reproducibility, you really can't have it without these key fundamental things. I think one of the other things that that's missing a little bit too is a focus on methods. So Jove is that's kind of the pillars that we stand on, is we focus on methods. That's what we do. And through our entire publication process, that's what we care about. It's what we invest our time and energy in doing, is really focusing on that. So I think that authors need to have all of the tools available to them in order to reproduce work. I mean, science is hard, let's be honest, how many times have you run the same protocol 3 times in a row and you don't know why it's failing, right? Like, that was my entire PhD. The entire thing from start to finish was all like that. And I'm sure I am not the only one in this webinar who had that experience. But that's one of the things, is I think that we really need to make efforts as publishers to really try and focus on that. And I think that that's something that's really important. And I think that all of these strategies are attempts at doing that. And I think it really does come down to a couple key things. You have to have the information available to you, and you have to know how to do the protocol. You have to know how the authors actually performed the experiments, because if you don't know that, then you really can't, it's not the same. You don't have the necessary information to be able to replicate that. And instead, you end up doing trial and error. And how many times have we all sat at a lab bench with the error part? Lots of work on the trial part, but so that's kind of our view from it, from a video publication perspective, is that we really care about the materials, we care about the methods, we care about the data.
Great. So we did have some questions from the audience. So one question was, could you please share the information about the persistent identifier again? So I think that might have been a question about identifiers.org. Is there another one that I should share? I'll put it in the chat.
No, I think that probably was it. There's Scolex.org, there's identifiers.org, and I mean, I know we're running at the end of the conversation, but I just want to say I really agree with what Justin just said. It's the holistic information. It is reagents, it's data, it is the protocol, it's the method or the workflow, and actually having all that combined actually is what matters. And my concluding remark is really that it's not so much the technical part of it, although I did start with that with those initiatives because that helps, but it's very much making it simple for authors and telling them, look, your paper will just get more read, more cited more, and get more attention if you share the method, protocol, and underlying data. And that is a message that they usually don't hear enough. And if we publishers do that all the same way, that actually is really powerful, and that will increase the uptake to the critical point that Anita cares about so much, the 10%. And we've seen it going up in terms of author uptake from 7% 5 years ago to more than 15% now, and I think that's only a starting point.
Great.
Yeah, I agree.
Okay.
And the other one, if you want to look at identifiers.org/RRID, that's where you can actually find a lot of the information from the RRID initiative on those identifiers. But there's lots of identifiers that you may want to link to, and so it's great to have you guys sort of representing the notion that we have a lot of these resources, and I think a lot of the big databases are really still underused. They're funded by large funding agencies, and I think they really want to be linked to. They want to have that impact. They want to increase their coverage, and I think that if we see ourselves as a large community, this is absolutely a wonderful way to go in terms of linking data, but I fully agree. It's not the end of the story. The methods are clearly extraordinarily important, and they should not be, second-class citizens any longer. So thank you for, to all of the people on this call.
Yes, and thank you, Anita. Thank you, Walter, and thank you, Justin. I'll turn it back over to Jeff.
Great, thank you all. Thank you again to Anita and Justin and Walter, and of course to Jennifer for moderating this excellent discussion. Thank you to our audience for your questions. Once again, please do take some time to respond to the webinar evaluation that you'll receive an email. Your feedback really helps us to continually improve this webinar program. Please also join us for the next SSP webinar, Being Author Friendly, 11 a.m. Eastern Time on Tuesday, October 16th. With that, we are concluded. Thank you once again.
Thank you.
Thank you.
Thanks.
Thank you.